Okay, everybody, this is going to be a quick and easy one. We're going to be talking about nucleic acids. So the only shtick that you really needed to go know about are like 5 major differences between our different groups of nucleic acids. You go ahead and take a look at the essential knowledge as I go along with it. Literally all it is that we're really going to be focusing on this is just talking about this section right here. Okay, if you can identify each of these things in here, you are going to be good to go. Okay, it's basically just talking about how DNA and RNA have the parts. You have the 5' and 3' regions, which we've already discussed about. You know, it's like everything you're already familiar with. So we're going to make sure that this is a quick one. It's not going to be half an hour. Hopefully we get this done within like less than 10 minutes. Okay, the first thing that we're going to go ahead and take a look at is some of the similarities. Okay, so we're talking about the similarities. So what are the things that are similar between these guys? Easy. They got a phosphate group, they have some kind of sugar, they have some kind of nitrogen base. No matter what, they have all these 3 things, right? This is what is unique about them, right? So they have the same components. They all run 5' to 3'. So remember, right, how you have like this 3' attached to the 5', 3, 5, 3, 5, 3, 5, right? They do the exact same thing, right? So that's the DNA side, right? And then we go take a look, 3, 5, 3, 5, 3, 5, etc. Right? We're the same thing, okay? And then there is going to be some kind of base pair, right? There's going to be some kind of bases that we're going to end up having, right? Okay. Let's go ahead and talk about the differences. RNA and DNA. RNA stands for ribose nucleic acid, ribonucleic acid. DNA stands for deoxyribose nucleic acid, or nucleic acids, deoxyribonucleic acids. In the case of it, right, ribose, we are seeing it that there's going to be an OH group right down here on the 2 position of our nucleic acid. And we go take a look over here, we have an H, right? Deoxy means that you have removed an oxygen. Super simple, right? So that is one key determining factor to be able to go and take a look. What we can end up doing is also getting the chemical formula, right? So we go look at the counts. We go 1, 2, 3, 4, 5 carbons. So that'd be C5, right? And we go and take a look at oxygens. 1, 2, 3, 4, O4. And then hydrogens. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, right? Oops, sorry, it should be like an H10, right? All right, so it's about that 1 to 2 to 1 ratio. All right, I'm gonna go ahead and just pull it over to the blue real fast and take a look, right? So same thing. So we got 1, 2, 3, 4, 5 carbons, C5. 1, 2, 3, 4, 5. 5 oxygens, and the hydrogens 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Right? So just giving you a little bit of a heads up, really the only difference right here is just we're looking at this oxygen right here, deoxyribose. Okay. Your next big difference. DNA contains thiamine. This is thiamine right here. So when it comes down to the base pairing, right, A will pair with T, adenine and thymine, right? We're going to have 2 hydrogen bonds, right? 2 hydrogen bonds when we're doing that twisted ladder. And G will pair with C. That's the 3 hydrogen bonds, right? In the case of your RNA, you have uracil. This is uracil. Now, RNA, for the most part, we're not going to go take a look at some crazy scenarios. We're just going to say the generally. A no longer pairs with T, it's going to pair with U. And G is going to go pair up with C. Right? Again, 2 hydrogen bonds here, 3 hydrogen bonds with these guys, right? 3 hydrogen bonds. Nothing is different. All that we're doing is just changing the nitrogenous base. Easy. We can go take a look on how they are similar, how they are different. Literally, all we're looking at is this guy right here and this guy right here. It just has an extra methyl group on top of it, CH3. That's the only thing that makes it different. Okay. When it comes down to your DNA, go back to blue. DNA is usually double-stranded. Now, I want to key in usually real fast because you're going to find exceptions where you can find single-stranded DNA molecules. It also says the same thing with RNA, right? If you go take a look, it says RNA usually single-stranded. And there's some cases where you might have RNA that might be double-stranded. For the case of this, we're not going to go worry about the exceptions. I don't want you to worry about the exceptions. We're just going to focus on this. Okay. When it comes to your DNA, it is usually double-stranded, which means that you have, right, here's your DNA molecule, and there's your DNA, right? Double helix with all this little nonsense going on in there. Perfect. That's what we mean. You got 1, 2 strands. You're double-stranded. Checks out. In the case of your RNA, it's just going to be 1 strand. So basically, it's going to look something like this, right? Right? There's no other side, so it's just 1 strand. Usually single-stranded. I'm telling you, this is a very, very simple one. This is showing you about it normally being single-stranded, right? But it can double up on itself. This right here is showing you how we can end up making some molecules. Okay, in the case right here, we can see how it bunches up and makes some double-strandedness, okay? But generally what it is normally a single-stranded molecule, okay? Again, RNA is usually single-stranded, okay? DNA is going to be anti-parallel. I know I've already mentioned this before. In the case of anti-parallel just refers to that you have 5' over here, 3' over here, right? And we are moving in this direction. We're going to have 5' over here, 3' over here. It's going to move in that direction. We already mentioned it before. It's basically because we want to make sure that each of these hydrogen bonds are actually forming and they are not designed to fold with each other if you have 2 lines both going 5 to 3. This just does not work. So instead, what we do is that we'll have one side go 5, 3, and this side will go 5, 3. And that's what we mean by anti-parallel. That's it. Just letting you know. Your nucleic acid for RNA and DNA, your sugar, the bases, single-stranded versus double-stranded. That's pretty much it, everybody. All right. That's literally it for this section. The only other thing that if you want to kind of keep in mind is just keep in mind that DNA will have DNA, right? And typically we just see it as a DNA, right? As that double-stranded. If you want to add one more guy into this, we can talk about how you have mRNA, tRNA, and then rRNA. And each of these does something different for protein for function, right? This is messenger. This is when you'd use the codon wheel, right? And it was like separate out. It was like A, U, GC, right? And then from there you'd end up going like A, U, G, C, right? And then each of those end up going out there, right? That's just basically this A, U, U, G, C, G. Like this is the thing that goes to the ribosome, right? That's the mRNA right there. Your tRNA are these little tiny molecules, right? We actually saw this earlier, right? They fold in on each other. They deliver the amino acid to the ribosome. They are the transfer. And finally, you have rRNA, which is ribosomal. It just helps build the ribosome. That's it, everybody. Hopefully, you'll go ahead and rest and relax. And at this point, you can start to work on your awesome handy dandy study guide, right? So again, keep in mind 1 and 1.3, right? You already should have gotten that one done. On the back side of that paper, you can now do your 1.4 to 1.6. Really, this is relatively simple. I wouldn't be surprised if you need to put anything in for 1.6. I would definitely focus a lot more on our proteins and the structures and stuff like that. And we'll spend some time going through it. That's all I got for you folks. Take care, enjoy, study up well, and I will see you soon.